Hello, my name is Scott McGuire. I'm a postdoctoral researcher here at Coventry University. My research looks at the acute physiological responses of hemodialysis and exercise in end-stage renal disease. Today I'm going to be talking about our most recent study, Cardiopulmonary and Metabolic Physiology during hemodialysis, inter- and intradialytic exercise. The use of inter- and intradialytic exercise is gathering strong evidence for the use with end-stage renal disease patients, with positive changes in aerobic capacity, cardiovascular function, and even improving solute removal. However, few studies have fully explored the physiological differences between exercise modalities, with most studies highlighting adherence and feasibility as key considerations when prescribing inter- or intradialytic exercise. This is problematic for the reasons that many studies have identified altered physiological responses when undergoing hemodialysis treatment, ranging from debilitating symptoms including post-dialytic fatigue to more serious complications of increased hypoxia, arrhythmias, and cardiac stunning. In the current study, we investigated cardiovascular and ventilatory changes to standard hemodialysis, inter- and intradialytic exercise. Both inter- and intradialytic exercise was individually prescribed. This was based on an equivalent workload to that of 90% of the anaerobic threshold achieved during a cardiopulmonary exercise test. The exercise duration was 30 minutes, and for the intradialytic exercise condition was commenced after 60 minutes of hemodialysis treatment. Outcome measures for the study included cardiac output, stroke volume, heart rate, blood pressure. We also looked at ventilatory gas exchange, which comprised of VO2, VCO2, respiratory exchange ratio, minute ventilation, and also arterial venous O2 difference. When considering cardiovascular responses, few changes were present between exercise conditions. Only cardiac output was identified to decrease over time, typical of hemodialysis. However, analysis of ventilatory gas exchange showed some very interesting results when comparing inter- and intradialytic exercise, with a respiratory exchange ratio above 1 during and post-exercise, a blunted minute ventilation, and increased VCO2 for the intradialytic exercise condition. When considering hemodialysis in isolation, we identified decreasing minute ventilation, VO2, VCO2, and arterial venous O2 difference. However, intradialytic exercise did appear to partly normalize arterial venous O2 difference later into hemodialysis treatment compared to standard hemodialysis. Mechanisms for the altered ventilatory response with hemodialysis may be explained by the leftward shift in the oxyhemoglobin dissociation curve, whereby use of a buffer, typically bicarbonate during hemodialysis to correct acidosis, may induce acute alkalemia and impair gas exchange at the tissue level, whilst also suppressing ventilatory drive. This mechanism would explain decreasing VO2, minute ventilation, arterial venous O2 difference, and increasing RER in the current study. This mechanism may also explain why increasing CO2 production with exercise may partly normalize arterial venous O2 difference. However, future studies are required to explore this mechanism further. To summarize, hemodialysis is associated with abnormal cardiopulmonary physiology, evidenced in our study by increased respiratory exchange ratio, blunted minute ventilation, and impaired O2 uptake and extraction. These perturbations likely also contribute to the altered acute physiological response observed during intradialytic compared to interdialytic exercise. Nevertheless, participants completed 30 minutes of intradialytic cycle ergometry at an intensity above the ventilatory anaerobic threshold with no significant adverse events. We hope these data may be useful for clinical and exercise professionals when prescribing exercise for end-stage renal disease patients and help better understand the acute cardiorespiratory changes that occur with hemodialysis treatment.